name is KaraFincham, and my biotechnology project is on DNA extraction. DNA extraction is the removal of deoxyribonucleic acid from a cell or a virus. Johann Friedrich Meister was the first person to extract DNA from a cell, and he was born on August 13, 1844, and he died on August 26, 1895. He actually discovered DNA on accident because he was just trying to extract a nucleus from a cell, and he ended up just extracting DNA instead. This is the process that it goes through.
This is the problem.
Okay. So at first the DNA is broken apart, and then it's used for multiple things such as genetic testing or body identification and forensic analysis, which is what this is showing. It's broken out from a cell and broken from the chromosome that's in the nucleus. This is just showing the DNA. To do this, most often, if they don't take a blood sample, they take a swab from your cheek and take a cheek cell and use it. This is just showing that. These are all the instruments that are used. It's a warm bath water, microcentrifuge, micropipettes, sample tubes, and a bunch of different solutions, which will be shown throughout the process. Okay, so at first you have to take a buccal swab and take a cheek swab, and then it is cut off and put into a test tube for sample. And this is just showing how it picks up the cheek swabs. After that, a lysis solution is added to the tube, and it is then put into, it's then placed into the warm water so that the proteins will separate and everything will break apart and the DNA will break out of the cell. This is showing how it breaks out of the chromosome and out of the nucleus and then out of the cell itself. After that, concentrated salt solution is added, and this causes the DNA to clump together and the proteins to clump together to separate them. And as you can see, the DNA is the strings, and the proteins and the debris left over from the cell are the little pieces, and it separates them. After that, the tube is put into a centrifuge, and what this does, and you have to add a counteract weight of just plain water so that it doesn't get unbalanced. And the centrifuge spins it around fast enough to separate the debris and the proteins from the DNA completely so that it can be extracted. And this is the proteins and debris that's sunk into the bottom of the test tube. Next, the water and the proteins and the debris are separated by removing the water and the DNA into a different test tube. the test tube is once again put into a centrifuge to take the DNA to the bottom. And those are the strands of the DNA coming together.
Those final.
After the centrifuge is done, all of the DNA is clumped together in the bottom of the test tube. The water is then extracted and you have your DNA separated. Advantages to DNA extractions are an opportunity to study genes, test for genetic diseases in newborns so they can see if any child will have a disease or a mental defect. And you can also analyze forensic evidence, which helps because it's helped gotten a lot of people out of prison because they were falsely accused for crimes. Disadvantages include if a DNA sample is kept in a laboratory for a long period of time, it will degrade and start to break down the DNA, and then it will be unusable. There's also harsh extraction environments. Temperatures have to be fairly cool and stuff like that. And this is my bibliography.
Any questions?
Kara, where can I get that stuff at?
What?
Where can I get all that? I want to do that.
We presented for like 2 minutes.